So good evening. It's my pleasure to welcome you all to this webinar. And thank you all for joining us on this night. So tonight, IVPN NeuroPsychiatry, in collaboration with Biogen, is conducting this MS webinar under the title Generic versus Branded Disease-Modifying Drugs and Therapies, Does It Really Matter in Multiple Sclerosis? So I would like to welcome all attendees, neurologists, residents, fellows, pharmacists, and nurses, of course, and all other allied healthcare professionals joining us tonight from this region, from the MENA region. And I'm sure you're all interested in the field of neurology and specifically in MS. Of course, we would like to thank Biogen genuinely for sponsoring this educational activity. Joining us on the faculty tonight are Dr. Sarah Dallal, Dr. Maya Zainuddin, and Dr. Jaber Al-Khabouri. So we will have 3 talks, each chosen specifically to enhance your knowledge about this topic. And we will start with the first talk by Dr. Sarah Dallal. She will speak to us tonight under the title of Innovator versus Generic Drugs, Definitions and Regulatory Requirements. So let me start first by happily introducing Dr. Sarah Dallal. She's the president of the Emirates Medical Association's Emirates Health Economic Society. She's an MD, a medical doctor, and she graduated from the UAE University College of Medicine and Health Sciences. She completed her internal medicine residency in Dubai Health Authority, and she holds a master's in healthcare management from the Royal College of Surgeons Ireland, and also a double master's degree on health policy and health economics from the London School of Economics. She's currently holding a position of Senior Health Services Specialist in the Insurance Policy Development section in Dubai Health Insurance Corporation. Thank you, Dr. Sarah, for joining us tonight on this hopefully very enriching webinar. And I'd like to ask you to start by sharing your screen and starting your presentation, the first presentation of our night.
Thank you, Joelle, and thank you for the whole faculty for this kind introduction. I hope you can see my screen.
Yes, we can.
I would like also to thank everyone who have joined us right now to just really talk about or to really understand more about the neuropsychiatry or the neurology world and all the advances that comes in. I will have a disclaimer at the beginning. Yes, I'm a health economist, but I'm not a neurologist or working in the neurology field. So the first presentation will just be discussing how about innovator or branded drugs are different than generic drugs. There are certain definitions and regulatory requirements, and I hope that anyone who is having any kind of question to drop them in the Q&A box. Again, my name is Dr. Sarah Dallal. I'm the president of Emirates Health Economic Society. So as a disclosure, this session, as mentioned earlier, is sponsored by Biogen Management Services. I will go to the slides where we will discuss briefly a general introduction about what could be a generic and what is a substitution for definitions. And when it comes to generic versus an innovator, what does bioequivalence means? And how does the regulatory approval pathway may look different between a generic and an innovator? And what could be also classified as a substandard medicine? There are lots of misconceptions when it comes to generic versus branded drugs or innovators, and what could be also a substandard medicine. And at the end, we would like to also present a small or research article of a multi-criteria decision analysis as how to the UAE looked at what could be a choice of medication when it comes to the quality of the medication or other factors. So starting with the first slide on what is generic and why is generic different than an innovator, or how it may look different than an innovator. A generic, as defined by the WHO, is a pharmaceutical product usually intended to be interchangeable with an innovator product. So by default, when we are talking about that, so innovators usually come before a generic is placed. So generic comes after an innovator. So it is manufactured without a license from the innovator company and marketed after the expiry date of the patent or the exclusive rights. So when it comes to the life cycle of a drug, which will be presented a little bit later, an innovator, a generic comes after an innovator with being manufactured without an exclusive license. There are similarities that helps to demonstrate why generic and innovators can be used interchangeably, and this is called bioequivalence, which implies that a generic medicine works in the same way and provides the same clinical benefit as the brand name medicine. So there are three things that we have to think about when we are talking about generics and innovators. Generics basically have the same efficacy, identical efficacy. They have the same safety and they have the same quality to an innovator drug. So there are lots of misconceptions when it comes with generics. And nowadays, there is a new other entity called biosimilars, but they are not interchangeable. Generics exist as the chemical molecules is the same as an innovator. And biosimilars or biotherapeutics in general are totally different. Well, they're different in a different sense. They are not the same copy, but they are, let's say, sisters to the innovator drugs. So when we're talking about a chemical molecule, which could be an innovator or a generic, they're produced by a chemical process. And typically, they are not complicated when it comes to molecular weight. So they're having a low molecular weight. Their structure is well defined. I hope you can see the icon up there. They are quite well defined. And they are mostly process independent. So there is no process that could be dependent in. They are completely characterizable and their immunogenicity is totally stable. Whereas when we are talking about biotherapeutics or biosimilars, they are typically produced by living cell culture. So by default, we can say they are a bit more complex or their structure is quite heterogeneous, and therefore they have a higher molecular weight. And they are strongly dependent on a processes for manufacturing, and they are not entirely characterizable. So they are not the same, in fact. Whereas when it comes to immunogenicity, it's unstable. So these are the difference between generics and biosimilars. And since the introduction of biosimilars, many may preconceive that biosimilars are in fact generics, but in reality they are not generics at all. So when it comes to substitution and why is this factor a very important factor, which is the types of substitution. When it comes to medication and having one area being substituted to another area, we need to really clarify why is a generic different than a therapeutic substitution or a pharmaceutical substitution. So just before a while, we discussed that biotherapeutics are not similar to generics because they're different in structure and complexity and processes and immunogenicity. And even when it comes to substitutions, there are three factors that three areas that we have to think about. First, an innovator and a generic could be substituted based on the merit that they have the same active ingredients. They are the same molecule to think of. they are actually chemically identical in strength, concentration, dosage form, and route of administrations. They're basically the same molecule with all the same characteristics that comes with it from strength, dosage form, and route of administrations. But there are other also forms of substitutions. And these two other forms that we need to keep in mind are therapeutic and pharmaceutical. So when we're talking about a pharmaceutical substitution, it is a different dosage form or a different route of administration. So one may be in an IM form and one may be in a pill formage. There's pharmaceutically, there might be, there might be the same in terms of active ingredient, but the route of administration or the dosage itself might be different. So this is where the substitution may lie in. And there's also therapeutic substitution, which is a totally different chemical ingredient, but may have similar treatment effect. So the chemical ingredient may not be the same, but they might be producing the same treatment effect. And there are many examples to look at. Maybe when it comes to that, it could be like NSAIDs, non-steroidal anti-inflammatory medications, where there could be the same chemical ingredient, but with similar, where different chemical ingredient, but with similar treatment effect. Now, going to the third part, which is bioequivalence. Bioequivalence means basically the pharmacokinetic term, which is to assess the expected in vivo biological equivalence of two proprietary preparation of the same active ingredient. So if there is an active ingredient, there are two things that we need to think about, which is the bioavailability or the bioequivalence, which is the rate and extent to which a drug is delivered from its pharmaceutical form into general circulation. So it is more defined by Cmax, Tmax, and area under the curve. For two products to be bioequivalent, they should have a 90% confidence interval of the relative mean of the Cmax, which is the concentration max, and they fall within the 80% to 125% from between the generic to the reference brand when it comes to the area under the curve. For the regulatory authorities such as FDA, EMA, the European Medicine Agency, Health Canada, and Australia, they should have the same definition of bioequivalence, which is basically both have the same rate and extent to which a drug is delivered from its pharmaceutical form and how it behaves inside or in vivo to the general circulation and therefore the biological equivalence. So basically, a reference or a generic, they act similar when they are in vivo format. Now, going to the drug discovery phase, which was mentioned earlier, there's a very long process when a drug needs to be discovered. And as you can see, you start from preclinical trials and you end up with the clinical trials being, or the RCTs, years and years after the preclinical trials. And it goes through multiple phases, starting by phase 1, phase 2, phase 3 trials, where an innovator drugs submits its dossier or submits its evidences to the regulators to have them approved. Once the medication is have been tested on the long term and got the regulatory approval, in essence, once the patency of the drugs have, or the innovator drugs have been completed or fell off, then the generic drug falls in or kicks off, and it will be approved or granted by the regulator. So talking about the regulators are now specifying the Food and Drug Administration based in the USA drug approvals for new drug applications or application to ANDAs. There is no necessary when it comes to generic drugs or to new drug applications. There is no necessity to repeat animal studies or clinical studies or even bioavailability studies if the main reason behind the difference is cost between the brand and the generic. Once you just approve that the same chemical ingredient is there, there is only the submission of a bioequivalent study. So for any A and abbreviated new drug applications, which is generic medications, all what is required are bioequivalent studies to make sure that the drugs are working or are behaving similarly in efficacy, in effectiveness, in quality. Usually or typically, the FDA monitors all approved drug products, including generics, to make certain that medications at all levels of the supply chain, from active pharmaceutical ingredients, the APIs, to products being sold to consumers, are safe, effective, and of high quality. So this is a bit of a separation between an originator, which is the innovator, versus a generic when it comes to the review process requirements. We just mentioned that there's a bioequivalence that needs to be looked at, but when the bioequivalence will require animal studies, clinical studies, and bioavailability. But if you can see with what is the requirements from the new drugs or the originators to ANDA, there are not many differences except for bioequivalent studies that may be represented with or its counterpart for animal studies, clinical studies, and bioavailability, whereas the rest is the requirements typically for all the medication, which is labeling, having the pharmacology and toxicology, manufacturing, and other controls, including microbiology, inspection, and testing. So to gain an FDA approval of a generic medicine, and we are just, the slides may be talking about the FDA approvals, but it's quite similar to any other agencies. For an FDA approval to grant a generic or generic medication, they should have a certain qualifications that we just mentioned earlier regarding the bioequivalence. And we are just pinpointing them now again in a more bullet format to emphasize that FDA looks at the same active ingredient as the originator medicine. So there is an active ingredient to be really represented. They should be also identical in strength, dosage form, and route of administration. So they could be substituted under the generic category. They should have or the same use of indications. So there should be therapeutically, when it comes to that, they should be having the same use of indications. In addition, there should be some studies that looks at Cmax and the area under the curve to be equivalent according to the guidelines, and that is called bioequivalency. They should also meet the same batch requirements for identity, strength, purity, and quality. And purity is a very major factor also here, which under that will fall into other implications if this is not met. They should be manufactured under the same strict standards of FDA's good manufacturing practice regulations required for an originator product. So also the manufacturing should have the same standards. Now, when we are looking at other factors like the Orange Book, which is the reference to products with therapeutic equivalence evaluations, there are a list of things that could be looked at under when it comes to the Orange Book and the approved drug products with therapeutic equivalence. So this is a great reference to look at what are certain medications with their generics, rather, and this is a very good reference that could be used for multiple purposes. Now we're looking at the drug approval process when it comes to EMA. There is a couple of things or four processes that we need to think about, or four steps that needs to be looked at. First, before the submission of any evidences, any applicant should notify the agency of their intention to submit the application. And there is a waiting period, which is of around almost 18 months in advance. So once the agency will be notified, they will be knowing the eligibility request. So this is just to formalize the eligibility request. Thereafter, there are steps prior to the submission, which are legal requirements. And this will also fall until the exclusivity period of a certain product when it comes to innovator product. Thereafter, there is the submission process. And the submission process starts with the procedure of reviewing the dossier, while the company may be answering any kind of opinion that may come or arise from the committee. There are also, while we can say similar application processes or application requirements that were discussed earlier with the FDA. But with the EMA, there is a circulation of the draft itself of the application or the submission to all member states from the European Union for review. The timetable for generic application states that there is an inspection request to be adopted, and by day 90 of the centralized procedure. So the inspection is announced for the applicant, and within 10 days from the adoption of the inspection request, an inspection sector will go and look after that. So it's an inspection on ground to look, which is parallel to the review time. Thereafter, which is a post authorization, the safety variations and the variations related to the safety issue will be looked at, and also the quality problems, and if there is anything that may be looked at in terms of labeling and packaging. So there is a process to an application for a generic drug under the EMA umbrella or the European Medicine Agency. Now, when we are talking about that, there might be some arise factors when we are looking at what could be a substandard medicine. And for most general public, they may think a generic may not perform as well, or the quality may not look as well as an innovator. But substandard medicines are totally having a different definition or a distinct definition according to the WHO. And to the WHO, a substandard medicine are genuine medicines produced by manufacturers authorized by the National Medicine Regulatory Authority, which do not meet the quality specifications set for them by national standards. So we are not saying generics are substandards. It could be any form of medicines that are not meeting quality specifications. And for that, we need to look at these four let's say figures down there, which we are looking at manufacturing process defects, or even packaging designs that are not adequate. Also looking at pharmacological variability and stability, and impurities in the drug manufacturing itself. So for that, if they are not met, then it could be considered as substandard medicines. So how substandard medicine may look like, they can have risk on the clinical outcomes. So they may have an absence or low therapeutic effects, and they might give the patients adverse events reactions even. They are also having risks on patient-reported outcomes. So there will be also loss of confidence in the drug itself, And as you as maybe can be having a wrong perception or perceiving of the medication efficacy. There is also a third fold for substandard medicines, which is the risk on economic outcomes. So there are certain hidden costs. So switching to more costly products by certain healthcare providers who are not convinced with generic use. So these are really legitimate risks when substandard medicines are taken into considerations. And that's why inspections and quality and regulators really do look for the quality of the medications and really warranting that the same standards should be followed in order not to fall under the three factors. There is an economic factor, an economic burden where there's hidden costs, and there is also the clinical and patient factors that may also lead not only to the wrong perception of medication efficacy, but even relapses with patients and even loss of compliance. Usually, the EMA moves to suspend 100 generic drugs over flawed studies run by different things. So there are usually typically are flawed or bioequivalent. And this is an article that was just released a month ago, but these are due to flawed bioequivalence. And the substandard medications typically are more targeted within the generic drugs because of the patency itself, and maybe the standards are not met, but that doesn't mean substandard medications cannot be in any other forms. Now, we would like to finalize this after talking about generics and how generics may look different. And there is a certain requirements for generics. Now, we would like to take another form about how generics could be more adopted in certain contexts and in a recommendation for a pilot MCDA tool to support the value-based purchasing of generic medicines in the UAE, in which Dr. Mohammed Farghali and the team, including myself, have looked at how we can best use generic medicines using an MCDA tool, which is a multi-criteria decision analysis. So if you are a budget holder or you are or you are on the formulary and you would like to adopt a generic, there is a certain way of using this tool to really give you a decision on what could be the best use of that. And this is where value-based healthcare really relies on, because generics are available and there should be multiple considerations. So how was this development of the tool would help in policy decisions? So with development of the framework, there was a framework, and then after that framework, there were also virtual workshop that was done during the last 2 years when there was the pandemic going on. But there was a preparatory work where the virtual workshop took place, and there was a weighting criteria. There were lots of policymakers and industry and the rest came on one table, let's say on a virtual table, to rank what could be the most evidence framework for off-patent pharmaceutical products when it comes to generic. And based on that, they started weighting their preferences using the pilot tool. And this pilot tool would support policy decisions for off-patent medications. So this was the criteria. And just not to focus on the criteria on the ranking itself, but if you look at what are the criteria on one side of the table, which is looking at what could an added value service product could be, or even the pharmacovigilance, or maybe economic or outcomes that we just discussed earlier, or quality assurances and equivalences, or even the stability and drug formulation, or pharmaceutical acquisition and cost. So really, when it came to the UAE and in Dubai in particular, when having all of these thoughts and all of these key opinion leaders, the criteria was put to them, and there was actually a weight that was placed when it comes to off-patent medications and generic medications. And surprise, surprise, the number one factor was not price. In fact, it came at number 5. But what it came to, it's the first criterion is having clinical, real world clinical or economical outcomes as a decision factor. So what we are trying to say, or what we are trying to promote, that almost 20% of the weight of the decision weight will rely on the clinical or economical outcomes if available when it comes to real world evidence data. So if you have a generic and they have real world evidence data, so for the UAE, that will be considered as number 1 rather than the price itself. Number 2, and also this is of high importance, is the quality assurance of manufacturing. Although they have on the third factor, which is the equivalence to the reference original product, which has also a significant weight of almost 15%, there is also these two things. So data comes first, and then the equivalence, and then the quality assurance when it comes to the manufacturing side, to make sure that there are no impurities, there are nothing that could be looked at or thinking, and then looked at the reference product. Stability and drug formulation is by default comes with the dossier. So, but still with that, it ranked at number 4. And then the price came in. So we'd like to encourage all the participants to get today with us to take the similar example and look at MCDA. Yes, if 2 certain medications could be substituted, but there are certain other considerations that should be looked at. Price should not be the only certain way of thinking of things, but there are other areas that should be looked at and should be considered when it comes to off-patent medications. And with that, I would like to thank everyone. And I'm so happy that I'm with you today. And hopefully I could have added a bit of what could be generic and innovator drugs. Thank you so much.
Thank you, Dr. Sarah, for your informative talk, where you really delineated for us the differences between innovator or branded drugs and generic drugs as well. Before we take some questions, and of course, we urge everyone to write down their questions in the Q&A. We'd also like to remind our participants that we'll be sharing a survey to fill evaluation forms after each talk. So we'll share it in the chat box. So I believe I'll ask Hossam to do that. And then next, we'd like to start with some questions for Dr. Sarah. Sorry, I was just fixing my virtual background. So can we conclude, Dr. Sarah, that more outcomes or clinical outcomes research is needed to really ascertain the value of certain generics?
Yes, actually, I think everyone is looking at real world evidence data right now. And I think because as long as the medication showed its bioequivalence, generally there is not a lot of research or appetite for research in for the generic, but there is a big area that we have to think about. Even if a generic is been placed, still real world evidence data, whether it was clinical or even economical, can really makes a big sense when it comes to generic off-patent medications.
All right. And so also next, we'd like to ask you a little bit about, because we also understood that the manufacturing process of generics is important. Who really monitors, like what regulatory bodies are responsible for monitoring this process and assuring the quality of this process?
Definitely. If we're talking on a global level, there are, like we just mentioned, the EMA have inspectors around that is going around in their member states, and they're looking at it. But in each country and around the world, they have inspectors themselves, and they're looking at the generic itself. And even with the submission of the dossier, they may be asking for samples to make sure, and also to look at the manufacturing site if they have the same standards as the innovator. So there are lots of considerations that could be looked, and this could be from the regulator of the of patent medication in the country itself.
Okay, and I believe in the UAE this is also available, right?
Yes, it is available under the umbrella of Ministry of Health.
Okay. So also another question is, so how can we as healthcare professionals really identify what you named as substandard medicines, and thus advise our patients accordingly?
Okay, definitely. That is a great question. When it comes to substandard medication, even if you're looking at it, you may not know if it's generic or not, but there is first of all, there is a big role from the regulator to ascertain and make sure that the medications are meeting the bioequivalence of an innovator. But at any point, if you are seeing your patients are not really reporting an outcome, and this doesn't have to be with substandard. If you're thinking that the patients are not benefiting from a certain drug, whether it was generic or non-generic, then substituting them to the best drug to save them from pain, agony, or even to have them in a better health outcome is the way to go. But in the same time, there are lots of multiple challenges that you can also report if you're seeing it multiple in a multiple fashion, or if you're seeing a certain batch, specifically if you're in a clinical pharmacy or anything that you are holding, please report it up to the pharmacovigilance and also to the regulators in your own facility that there might be something wrong with the certain medication at question.
Perfect. And probably our last question, what is the size of the sample that generates reliable real-world evidence data?
Oh, that's a 1 million dirham question, 1 million dirham. Now, let's look at it in a very in a very substantial way. It depends from a disease to a disease, a drug to a drug to a drug. So it's all I can say, the more the merrier. Whatever you can get as data is best. But what is more important is not the number, the quality of the data itself. Real world evidence data nowadays have some standards themselves to get them. So it's not only just capturing the data, but also getting the good data to really inform decision.
Perfect. That's a perfect point, I think, to wrap up. I'd like to remind the participants to fill the evaluation form shared in the chat box. And I believe we're going to end here with Dr. Sarah. Thank you so much, really, for a very enriching talk. And we're looking forward to have you in future webinars as well. Thank you.
Thank you, Ms. Joelle, and thank you, everyone.
All right, so let's move to our next talk of this night, of this webinar. So our next talk will be given by Dr. Maya Zainuddin. She will discuss the generic disease-modifying therapies in MS, their availability, their safety, and their quality. And of course, I'm sure many of you know Dr. Maya, but it's always my pleasure to introduce her as a colleague and a dear friend. So she's a Lebanese senior clinical pharmacist specialized in neurology and MS specifically. She completed several internships at major hospital and community pharmacies in Lebanon and the US, and graduated from the Lebanese American University with a Doctor of Pharmacy degree with distinction in June 2011. She was also the winner of the Best Clinician and the Positive Attitude Award. In August 2015, she completed and successfully passed the multiple sclerosis specialist examination and became the first MS certified pharmacist in Lebanon and the entire region. So from 2011 to 2020, Dr. Maya was the clinical pharmacist at the Multiple Sclerosis Center at AUBMC, which was the first multidisciplinary center, MS center in the region. In December 2015, she was also appointed as the Executive Secretary of the Middle East North Africa Committee for Treatment and Research in MS, MENACTRIMS. She is an active researcher. She's involved in several international clinical trials in the field of MS specifically, and she's a member of several national and international professional organizations. Maya is also the fund coordinator of the National Multiple Sclerosis Society of Lebanon, where she helps patients in these difficult economic times to be able to really stay on therapy and achieve good outcomes. She's the leader of the NeuroPsychiatry listserv at IVPN, so she's behind all this great work you're seeing today and on all our webinars. And of course, as a continuous learner, she's also currently a PhD candidate at the University of Limoges in France. So Dr. Maya, thank you so much for really diving into MS and disease-modifying therapies, generic versus branded in MS. And I'd like you to start, and I will remind our attendees that we will take questions right after your session as well.
Thank you, Joelle, for this great introduction. And before starting my lecture, let me thank Biogen for inviting me to give this talk about the use of generic disease-modifying drug for MS, their safety, their availability, what is available, what kind of generic DMT we currently have in the market, and the quality of this drug. So Dr. Sarah made my job easier by giving us a very good and excellent introduction, general introduction about the generic drugs in general and the regulatory process for being approved and used in many diseases, not only in multiple sclerosis. So here are my disclosures. And let us have a look on what do we have as disease-modifying drug in MS. So as of 2021, there are now 19 plus approved branded disease-modifying therapies for multiple sclerosis in the US, but also in Europe that are FDA and EMA approved for the treatment of MS. And as you can see, before 2011, there were very limited options for the treatment of MS, and the treatment of MS was mainly limited to injectable drugs, which are low-efficacy disease-modifying drug, interferon, glatiramere, and then natalizumab was introduced in 2004. It was the first monoclonal antibody for the treatment of MS. It's one of the highest efficacy disease-modifying drug. Then in 2011, the first oral DMT was approved, and starting 2011, you can say that the era of DMTs started, and we are having every year one or two or many disease-modifying drugs that are approved. And as you can see now, we have many options. Some of these drugs are oral therapies, other are injectables, other are intravenous drug given in the hospital. They, of course, differ by the mechanism of action. Some are immunosuppressive drug, other immunomodulator, and even within immunosuppressive drug, we have different different mechanism of action, and each one target different immune cells. And also they differed by their efficacy. So we have low efficacy disease modifying drug, and we have high efficacy disease modifying therapies. And we have many other drugs that are currently in the pipeline being studied, and probably they will be approved in the upcoming years for the treatment of MS. So really we have a diverse variety of DMT that are safe, effective, approved for the treatment of MS, and those drugs were approved based on, of course, clinical trials and follow all the FDA requirement for any drug to be approved. Let us have a look on what do we have currently on our market in the Middle East and North Africa region. So as you can see, this table is the kind of a very busy table. So here are the drugs that are FDA or EMEA approved for the treatment of MS. So these are the unlabeled drugs that has a label for MS. In white, those are general immunosuppressive drugs. Those were used in MS previously, traditionally, when we didn't have these diverse options. So whenever a patient used to fail injectable approved DMT, we didn't have any other option to treat this patient. So physician or neurologists tend to use off-label immunosuppressive drug like azoxystrobin, mycophenolate, mofetil, fludarabine, chemotherapy, cyclophosphamide, this is another chemotherapy drug, methotrexate, mitoxantrone. So either immunosuppressive or chemotherapy drugs that are usually approved for other condition like renal transplant, cancer, but we use them off-label for the treatment of MS. So those are the medication in white. And these, among the unlabeled DMT, they are, you can divide them into or categorize them according to their efficacy. So we have the high efficacy drug, alemtuzumab, natalizumab, ocrelizumab, rituximab. We have the moderate efficacy, those are the oral treatment, fingolimod, siponimod, dimethyl fumarate, and we have the injectable and teriflunomide, which are the low efficacy DMT. And here are the MENA 16, Middle East and North Africa region. So if you look at this table, you can notice that unfortunately, despite that we are in 2022, we have 2 countries in the MENA region that didn't have any approved DMT on the market for the treatment of MS, and those are Syria and Sudan. So we don't have any DMT option for patient there, approved DMT option. So only off-label DMTs are approved. And if you look also for the high efficacy DMT, 20% of the MENA region country, they didn't have any high efficacy DMT. Those are alemtuzumab, natalizumab again, and ocrelizumab and rituximab. So in those countries, if a patient fail low or moderate efficacy DMT, he doesn't have any high efficacy option to shift to. So this is also another issue and another important thing to know. So really the distribution of DMT among the different countries in the MENA region vary significantly, and this is because the MENA region is a very political and economically, the countries differ economically and politically, and we have a region where they are a region of instability due to war or conflict. And if you can see, for example, Kuwait is the only country where we have approval of siponimod, and the Gulf countries are the countries who have the highest number of approved DMTs available, and this is related to the high GDP or high economic status in these countries. So because the approved DMT, they are not available in many of the MENA region country. In some countries, they tend to use generic disease modifying drug, and some regulatory approval, some regulatory authority actually approved follow-on or generic DMT in half of the MENA country. So 50% of the MENA country, we have approval for a generic disease modifying drug, and those are follow-on teriflunomide, generic for interferon beta 1b, 1a, generic for glatiramer acetate, generic for fingolimod, dimethyl fumarate. So those are the DMT for which a generic drug is available, and they are available in 50% of our MENA countries. But you should keep in mind that many low-income and emergent countries, they do not have value-based or market-based price strategy to regulate a competitive market. So furthermore, pharmaceutical manufacturers of generic drugs have more flexible internal rule of compliance and less bureaucracy compared to brand name companies that are usually obliged to report to their head offices. So here we cannot we cannot guarantee that in those countries where we have low economic condition, where we have unstable political, economic situation, and we have really a need for a DMT, that in those countries, the regulatory authority, they are stringent or have stringent rule. And also the manufacturer of the generic DMT also usually, they don't have the same stringent regulation as the multi-international pharma company that usually manufacture the branded disease-modifying drug. So in the MS, in multiple sclerosis disease, we have very few approved generic DMT by the EMEA and FDA compared to other disease area where we have more generic DMTs approved. And this is because due to the inability to perform pharmacokinetic study for many of the DMTs, also because of the insensitivity of the clinical measure typically used in pivotal trials. So we don't know what is the best clinical endpoint that we use to measure. Is it relapse? Is it MRI? Is it disability progression? Also lacked of validated non-imaging biomarker that relate to efficacy. So until today, we don't have biomarker for efficacy in multiple sclerosis, and that's make the evaluation of efficacy of generic DMT more difficult for multiple sclerosis. And also keep in mind that to study a generic DMT in MS, this need a clinical trial and involvement of patient, and you need really, it's very critical or very unethical sometimes to enroll MS patient in sub-study, especially that multiple sclerosis is an irreversible disease and it's a disabling one. So whenever it's unlike, for example, hypertension, if a patient try a generic antihypertensive drug and he developed high blood pressure, this is reversible. He can just change the medication. He can just take another antihypertensive drug that will lower his blood pressure. However, in MS, if you try any generic DMT, and the effect will be shown over time. So any DMT will take 4 to 6 months to kick in and to show efficacy. So, and by this time, if this ability develops, then this will be, we will be at an irreversible stage. So really also studying generic disease modifying drug through pivotal clinical trial is also uneasy, and it's difficult because of this ethical issue and because of the nature of the disease that is irreversible, and it's a disabling disease that can lead to irreversible physical and cognitive disability. So let us look, Dr. Sara explained to us the regulatory requirement for generic and branded drugs to be approved. Let us look for DMTs, for innovator and generic DMT regulatory requirements. So for brand disease modifying drug, usually 2 clinical trials are required to confirm safety and efficacy, and usually you need to prove superiority to placebo, and nowadays we have more clinical trials that demonstrate superiority to a low efficacy disease modifying drug like teriflunomide or interferon. So but we need to demonstrate superiority either to placebo or to comparative active disease modifying drug in order to be approved. However, small molecule generic can be verified as being safe and effective by only showing either pharmaceutical equivalence, so it's the same active ingredient, purity, strength, dosage, and route of administration, and bioequivalence, so similar rate and extent of absorption, providing the same exposure, and Dr. Sara elaborated a lot in this area of bioequivalence. So as a result, only small trials comparing the pharmacokinetic of the generic and the brand is required. So here we have a small study with a small number of patients focusing only on pharmacokinetics endpoint, not like clinical trial, phase 3 clinical trial that usually enroll thousands of patients. We have a very big number of patients, and we have many clinical and efficacy, safety and efficacy clinical endpoints. So it's very important also because we hear from our patient that I can use this drug because it has the similar active ingredient of another drug. So this is also a misconception in our community, that the quality parameter, not only of the active ingredient, but also of the excipients, are important for patient outcome, and all those should be controlled. So it's not about a medication having the same active ingredient, but also about the quality of the excipient and the quality of the active ingredient. And this is because some excipients significantly affect the bioavailability of the drug and modify the product at shelf life. For example, you can see here, for example, the packaging, the primary packaging, it will affect the material composition and can affect the stability and therefore the safety. Also, the manufacturing process, the quality by design, the content uniformity, the batch-to-batch variability, and all the manufacturing practices, those can affect the dissolution, and the solution will affect the efficacy and the safety of the drug. They can affect the dosage, the efficacy, and the purity and sterility, which affect the safety. Also, the dissolution, the type of the excipient, what kind of excipient is used in this drug will significantly affect the bioavailability, and bioavailability, as you know, is a key important factor for the efficacy and safety of any medication, not only disease-modifying drug. So usually approval of the current DFT for relapsing remitting were based on demonstrated benefit on relapse rate. So if you look at the clinical trials for disease-modifying drug in MS, the primary endpoint is the relapse rate or the annualized relapse rate. And because these measures are relatively insensitive, MS pivotal trial usually lasted 24 to 36 months and enroll several hundreds of patients and even thousands of patients. And MRI measure of lesion activity are usually more sensitive than the clinical parameter. However, they are accepted by regulatory agency only as supporting secondary endpoint of a new drug. So usually when a new DMT needs to be approved, it needs to demonstrate superiority in terms of efficacy, and the efficacy endpoint are usually divided into primary endpoint, which are the relapse rate. The annualized relapse rate is usually the primary endpoint in most of the study, and the secondary endpoint will be the imaging parameter, which is the appearance of a new T1, T2, or enhancing lesion on brain MRI, and the disability progression, which is measured by EDS. And to evaluate whether lesion activity could serve as a surrogate for relapse in MS trial, a meta-analysis was performed for 23 clinical trials, and that demonstrated a strong correlation between treatment effect on MRI lesion activity and clinical relapses at the trial level. So MRI lesion activity was considered an appropriate primary endpoint for the approval, or in comparing generic versus brand DMT with established effect on MRI lesion activity and relapses. So those are usually the equivalence trial design, or is the gold standard design for approval of generic disease-modifying drugs. So although a non-inferiority study design has a more statistical power in general, in situation where a clinical trial is required, an equivalency design is preferred, and this equivalence trial should show that the generic alternative is neither less or nor more effective than the reference product to a meaningful degree. And in general, the equivalence limit are 80 to 125%. Those are the acceptable equivalence limit for a particular parameter. However, unfortunately, the sponsor and the regulatory agency can even negotiate those actual limits. So really, we don't have stringent rules for the approval of generic DMTs. This is the example for the approval of the generic Copaxone. Copaxone is one of the first approved disease-modifying drug for MS. It's glatiramer acetate. It's an injectable 20 milligram given subcutaneously daily. So the patency of Copaxone ended in 2014, and the EMEA viewed Copaxone as a complex generic, and they advised Pfizer to perform a clinical animal trial to confirm the equivalent safety, efficacy, and tolerability in patients with MS, and this led to the GATE trial. It was a multi-center, randomized, double-blind, active, and placebo-controlled phase 3 trial. It is an equivalency study to demonstrate that generic Copaxone is equivalent to the brand one. Eligible patients were randomized either to generic glatiramer acetate, so 353 patients are enrolled, brand generic glatiramer acetate, 357, or placebo, 84. Administered the subcutaneous injection for 9 months. As I mentioned, we cannot perform studies in MS for a short period of time because DMT needs 4 to 6 months to kick in. And the result demonstrated that generic and brand, they have equivalent efficacy, tolerability, and safety over 9 months. And based on this, the generic of Copaxone was approved and introduced into the market. However, the study, if you look into it, it has many limitations. The major limitations were that the study was short, so 9 months is usually not enough. If the drug usually take 4 to 6 months to kick in, so 9 months wouldn't be enough to assess its efficacy. And also the small number of patients in the placebo group, so this will lead to a low statistical power for the primary endpoint. So this GATE trial provided a general template for equivalency trial of generic DMT, which showed that, again, we don't have strict rules and policies and regulation for the approval of generic disease-modifying drug in multiple sclerosis. So what are substandard studies? If a medication failed to meet the regulatory, which are the FDA, the EMA, WHO quality standards, such as the good manufacturing process and bioequivalency that Dr. Sarah Elab explained it, they are then defined as substandard copies, and this can pose serious risk to patients in terms of safety and efficacy. And as I mentioned to you previously, efficacy in MS is very critical because we are dealing with an irreversible disabling disease. So potentially non-controlled or different manufacturing process and excipient in non-proprietary drugs may result in poor batch-to-batch reproducibility and lower quality. And these are substandard non-proprietary copies of DMT that are immunomodulatory or immunosuppressive are of concerns to patients due to their possible untoward safety and lack of efficacy event, as I mentioned. And use of ineffective, poor quality, harmful medicine can cause therapeutic failure, disease exacerbation, resistance to medication, and sometimes can lead to death due to toxicity issues. So substandard medicine are prevalent in region where the health authority, they don't have a presence. So where the health authority, they do not enforce registration regulation that are stringent and that are enforced by the FDA, EMA, and WHO. So those are the countries they do not follow or they do not report to those big regulatory authorities. And globally, 15% of the medications produced in general are substandard, and the rate in Asia and Africa, so in our region, unfortunately is high. It's 40 to 50%. So 40 to 50% of the medication produced are substandard drugs, and they do not meet the good manufacturing process or the good bioequivalency properties. Here are examples of switching from proprietary to non-proprietary copy from other therapeutic area than MS. So the issue is critical in MS, but it's also a concern in other disease therapeutic area. In epilepsy, for example, there was reports of toxicity. Many studies showed the reports of toxicity, decreased efficacy. So we had a breakthrough epilepsy in patients with epilepsy who have switched to copies and have led to increased scrutiny by professional body to prevent or reduce the use of anti-epileptic copy medication. So it was a total failure whenever the patients shifted from branded anti-epileptic to copies, to non-proprietary copies, and led to therapeutic failure. Also, in renal transplant, a study of renal transplant patients in the US revealed higher total healthcare costs associated with generic cyclosporin A, and this is because of therapeutic failure that led to higher cost and hospitalization and need for a visit and management. So the cause of cost difference was higher rate of exposure of immunosuppressive in patients receiving generic cyclosporin A, in addition, as I mentioned, to suboptimal efficacy and therapeutic failure. So really, you should be careful when reading a trial comparing branded versus generic DMT and when evaluating a generic drug. Those are examples from multiple sclerosis, the clinical implication for substandard copy, beta interferon. So usually beta interferon, as I mentioned, they are one of the first disease-modifying drugs. Those are injectable, given every other day or weekly or 3 times per week. So we have different types of interferons, and they are used for relapsing remitting MS as first-line treatment. So those are low-efficacy drugs. We have many generic interferon in our region, mainly from Iran. We have Sinovac from Tehran, Iran, Gemfaxone from Switzerland. And also we have, those are the generic drug for Avonex, which is the brand name for Biogen, and Rebif, which is for Merck Serono. And those are available commercially in many countries. In Syria we have, in Iran we have, and in Lebanon we have, and in many other countries. And unfortunately, if you look at the clinical trial of Sinovac, it really shown very poor efficacy compared to the branded Avonex, with increased relapse rate, increased number of lesion and enhancing lesion on brain MRI, and increased disability progression compared to the branded drug. Also, Gemfaxone, which is the generic drug produced in Switzerland, it displayed an unfavorable tolerability, and a very high proportion of patients discontinued this medication compared to Rebif due to side effects, mainly flu-like symptoms, local skin reaction, and mood change, which are reported more frequently with the generic interferon. Also, biological studies on potency and molecular characteristic between innovator beta interferon and corresponding follow-on interferon from Mexico, Uruguay, and Iran. All these studies revealed substantial differences in biologic and immunological behavior, suggesting that these molecules, in fact, are not similar therapeutic agents, but they are totally different molecules. So they are really new drugs with different efficacy, which is lower than the branded efficacy, and they don't behave even at the biological and immunological level the same as the branded drug, and that's why we should be very careful. And multi-center pharmacological study in Latin America have shown reduced bioavailability of the generic beta interferon compared to the innovator. So we have lack of efficacy, unwanted or undesirable side effects. With the generic interferon, we have decrease in bioavailability, and really we have different immunologic effect in vitro for these medications. And despite that, they are being widely used in our MENA region country, either because lack of availability of the branded DMT, or because the regulatory authority of the country approved and give priority for these drugs, or due to cost, for example, and any other reasons. So this is another example of substandard copy. The experience with fingolimod. So fingolimod, which is Gilenya, the branded one, which is produced by Novartis. It's a sphingosine 1-phosphate receptor modulator that crosses the blood-brain barrier. And for medication that cross the blood-brain barrier and work into the CNS, the health authority usually requires the manufacturer to complete a specific additional clinical investigation other than the usually requested or required ones. And substandard fingolimod copy that do not undergo such an investigation may cause side effect problems for patients because of the mechanism of action of fingolimod and its effect into the nervous system. And the initial phase of the manufacturing process of fingolimod incorporate a chemical that has genotoxic property. As part of the quality assessment of proprietary fingolimod, genotoxic level should be below the threshold of toxicological concern, which is 1.5 microgram per day, is set in the guideline of the International Conference on Harmonization of Technical Requirements for Registration of Pharmaceutical for human use. And copies of fingolimod that become commercially available should follow all these proper legal approval and conform to the good manufacturing practice and other quality evaluation. So this is a comparison of the proprietary fingolimod, the branded fingolimod, compared to 11 fingolimod coffee that were produced. So the proprietary fingolimod was considered the reference, and it was again compared to other 11 non-proprietary fingolimod. So they look into difference in particle size, distribution, and level of impurities such as heavy level. And as you can see, for microparticle size and heavy metals, all the tested fingolimod copy were out of the specification by several fold magnitude. So they failed and they out of specification. And if you look at the percentage, for example, we have 73% out of the specification for the sulfated ash and 100% out of the specification for the heavy metal. And if you look at the percentage of impurities in these non-proprietary fingolimod, it is 46%, which is too high, because the impurity frequency or percentage allowed by the ICH technical requirement for registration of medication is only 1% and should not be above 1%. However, all the non-proprietary fingolimod has had a percentage of 46% of impurities, and this can lead to toxicity. This is another study from Argentina. Why Argentina? Because Argentina is one of the countries that had the most number of generic disease-modifying drug. Almost all disease-modifying drug, they have generic there. Unfortunately, they don't have regulatory requirement for the introduction of generic DMT in the market. So Argentina, they do not require bioequivalency study or other requirement for introduction of generic DMT into their market, and that's why we have a big number of generic DMT into their market. So this study, it is an observational one where they assess the role of or the impact of prescribing generic non-proprietary drug into the prevalence of therapeutic inertia in multiple sclerosis. So this was their objective, is to compare this prevalence of therapeutic inertia between branded and generic DMT. And for this, this was, by the way, a population-based survey comprising 117 neurologists, experts in MS. And they created something called generic brand score. And this is a score where if it is more than 1, this means there is a higher incidence of prescription of generic drug. And if it is less than 1, which means that we have more brand drug, or a higher number of brand drug prescription. And if you can look at the result, it showed that a higher number of T1 lesion was present or correlated with a higher prescription of generic DMTs. And whenever even this result was reproduced in the multivariate analysis, where they controlled for all the other confounders that could lead to therapeutic inefficacy and appearance of breakthrough disease and brain MRI in MS. And even when they controlled for all the confounders, still the prescription of generic DMT was associated with higher T1 lesion compared to generic DMT. And interestingly, in this study, it's not shown reflected in my slide. However, they also shown that there was no difference in the annual cost for the management of MS between the prescription of a generic DMT compared to a brand DMT. And this because Dr. Sarah mentioned, she mentioned this clearly, that when prescribing generic DMT, we have hidden costs. So it's not only the cost of the medication, and this is proved in the study from Argentina, where they shown that the prescription of generic DMT was associated with higher annual cost because patient had more relapses, more breakthrough disease. They had they had to be admitted more frequently. They had more a clinical visit, more ER visit, the need for steroid infusion, and this further increased the cost of treatment per year. So really, approving generic disease-modifying drug for multiple sclerosis, unlike other therapeutic area, is very delicate, very critical, and should be evaluated very carefully. And we should have standard regulations, especially in our MENA region, for approval of the generic DMT, because as we mentioned, it is an irreversible disease, and whenever the damage will be shown after 6 or 1 year of use of generic DMT, it will be too late to reverse the damage. So how can we prevent the use of these substandard copies in our region? We need to establish WHO EMEA standard regulation. We need to have well-trained evaluators and inspectors who will make sure that those regulations are well implemented. And this is the key for registration for only quality generic and biosimilars in our region. And also neurologists should play, and us as clinicians, we should play a major role in front of authority who might allow the use of substandard copy or generic DMT that do not meet the good manufacturing process and bioequivalency study, by always reporting side effects, any therapeutic failure with the use of generic DMT. So those are the take-home messages. So then the introduction of generic drug into the MS treatment landscape could reduce the medication cost, especially that DMT, as you know, are usually very expensive medication. And success in the development of a generic drug will depend on the ability to offer a similar, safe, and effective product at a cost saving. So this is a very nice theoretical thinking. However, as discussed, it is not straightforward to demonstrate this point for complex generic. Too relaxed requirement may jeopardize efficacy and patient safety and lead to toxicity and inefficacy and to disability. And too stringent requirement may create a disincentive to pharmaceutical companies. And MRI endpoint usually should be used as a surrogate marker and endpoint for clinical efficacy to make equivalence trial more feasible. And switching to a substandard alternative may increase the long-term healthcare cost and exposes patients to ineffective treatment. Just 2 more slides from my perspective and from a pharmacist perspective. MS patients should have access to safe and effective treatment, which might meet the high standard of proof for quality. And the development of generic is an important process in order to make DMT available for all people, not only for people or for county with high economic status. So treatment should be accessible and available for all patients. So it should be more widely available at a more affordable cost to health system and patient of MS. So from this perspective, we should encourage the production of generic drug. However, exchange between biological medication require adequate clinical monitoring, detailed record keeping for traceability, clear and balanced information, and consent by the patient. All treatment, proprietary, generic, biosimilar need to meet and be able to demonstrate stringent safety and efficacy data and be properly assessed by independent regulator. And we do not support the use of substandard medicine or copy that have not passed a stringent test for quality, efficacy, and safety. And from pharmacoeconomic perspective, not always a generic drug is more cost effective than a brand. So this is a major misconception among healthcare professionals. So usually the generic drug is cheaper. It's not always the case. So this is again in a study from Argentina. I told you why this study about generic DMT are always come from Argentina, because they have a big number of generic DMT, and because they do not require any bioequivalence study for the approval of generic drug. And if you look at in this table, the annual cost of MS treatment in Argentina did not differ between the generic and the brand name. If you take, for example, the example of dimethyl fumarate and for natalizumab, it is the same. It's almost 50 to 57,000 for generic, and it is 63 for brand name. So we don't have any significant difference, for example, for the generic of dimethyl fumarate. And this is because the cost of generic, as I mentioned, was similar or higher compared to brand name. And this is because of the hidden cost associated with the use of generic DMT, and could be related to safety issue and therapeutic failure that will lead hospitalization and doctor's visit. So it's not always using a generic drug is not always a reduction in the annual cost of treatment. Here, another study from the USA that compared the effect of the introduction of generic glatiramer acetate on the spending and use of drug for MS. And the ends you can see with the introduction of this generic glatiramer acetate, there was an immediate increase of $441 in the cost per prescription of the branded glatiramer acetate. And this is again because of the competition, and this will lead to higher cost on the healthcare system and on the patient. So generic may not have the same cost lowering power for specialty drug in MS compared to other therapeutic area, especially when other brand name drug are approved to treat a given disease before the first generic drug is approved. With this, I will end my presentation. I would like to take the opportunity to invite you all to our 7th Minectrim Congress that will take place in Cairo next November 11 to 12, and where we will be discuss all the updates in the treatment of MS. So you are all welcome to attend, and thank you for attending our webinar today.
Thank you so much, Dr. Maya. As always, a very packed and comprehensive presentation. So I would like to also remind the audience to fill the evaluation form that will be shared in the chat box in a minute. And so I believe you really clarified for us some misconceptions that we, as healthcare professionals, we might have when we think that generics are always more cost effective. I believe we really pinpointed those misconceptions and discussed them. So some comments from our audience. From Egypt, one of our attendees is telling us or informing us that cladribine and siponimod are now available. I believe this is in reference to one of your slides.
Yeah, so this study, Joelle, was performed last year. So last year we performed the survey, and usually the neurologists answer, or the regulatory authorities in each county answer based on the year before. So this is an assessment of the market in 2020. So of course now things are changed since 2 years, and we have the introduction of siponimod in many markets, not only in Egypt.
Definitely, especially in the MS world, things are changing every year. So this is a question that we maybe answered before, but if you can brush up on it, what are the regulatory bodies that are responsible for regulating the quality of generic drugs?
So usually the Ministry of Health in each country, they should have a department for approval of generic DMT. This is the regulatory body in each country that is usually responsible for the approval of all drugs, the generic and DMT. But for generic, we usually have a special department in the Ministry of Health in each country in order to check the bioequivalence study and if this generic drug meet all the GMP requirement before giving the pharma company, the approval to introduce it in the market.
And another attendee is asking, is there any clinical trial that compares Rebif and Avonex?
No, we don't have clinical trial that compare Rebif and Avonex.
Okay, I believe these are most of our questions. Just to wrap up, you really discussed the hidden costs that might be associated with generic drugs, and also, do you believe that the approval process is more complicated and rigorous for branded drugs versus generic, or do they both follow the same
No.
stringent approval processes?
No, for brand, as we discussed, for branded drug, for the approval of branded drug, it's the process is more complex, starting from the design of the clinical trial that should show superiority either to a placebo or to an active DMT comparator, and also the number of patients enrolled should be a big sample size and not a small sample size, and reaching all the bioequivalence study, the pharmacokinetic study, and the safety studies. However, for generic disease-modifying drug, only a small study include a small number of patients is enough, and focusing only on pharmacokinetic property of the drug and proving equivalency to a branded DMT is only required. So of course we have more stringent rules for the approval of branded DMT, and also usually branded DMT, they are introduced to the market by international pharma company that themselves also they have very strict and stringent regulation and compliance rules.
Okay, we'll end with two questions, if you can answer them for us quickly, so that we move to our next talk. Any clinical trial generics versus branded teriflunomide?
Not to my knowledge. We have clinical trial for dimethyl fumarate, but not to my knowledge for teriflunomide.
Okay, and why don't we use only innovative DMTs where generic is not available, like Tysabri, Lemtrada, and others, to give MS patient maximum benefit?
Yeah, we hope that we can do that, but unfortunately this is not feasible all the time, because those drugs that you mentioned that are drugs that are very, very, very expensive, and they might not be available in many of our countries, and if they are available, they are not always covered by insurance companies, and usually patients cannot afford their cost. They are very costly drugs.
Thank you so much, Dr. Maya. We will move to our next talk by Dr. Jaber. Thank you. So thank you, Dr. Jaber, for your patience. I know we passed the time a little bit, but the discussion was interesting. So we will end with the third talk by Dr. Jaber Al-Khabouri. So Dr. Jaber is a senior consultant neurologist and deputy head of neurology in Khula Hospital, Muscat, Oman. He graduated from Sultan Qaboos University Hospital Medical School in 1994. He was a distinguished student and he received the Dean's Award of Achievement. His medical training was in Royal Hospital, and he received also the award of Oman Medical Specialty Board for the best residents. He passed his MRCP diploma in 1997 from Glasgow, and his neurology training was subsequently in London from 1998 to 2003 as a specialist visiting registrar. He returned from England in 2004, where he headed the neurology department for several years. He has training in all neurological disorders, but his main interest is in MS, headaches also, and vascular pathology. He is trained in Botox, interpreting EEG and EMG. His research track really revolves around oral treatment for MS and cannabis use in MS as well. And he has published several papers on MS in Oman. He is currently running the MS clinic, and today he will be talking to us about the Omani experience of the safety and efficacy of generic MS therapies. Thank you, Dr. Jaber, for joining us tonight.
Thank you, Joelle, for this nice introduction to my talk, and I thank the previous speakers for setting the scene for me, and above all, we thank Biogen for sponsoring this, and obviously the audience who kindly has come all through to listen to us. And this is a teaching purpose, or a teaching. Now, you have listened to the previous speakers, who actually, one is the health economic and the second is a prominent pharmacist and a colleague. Now, I'm going to give you a physician perspective of this, because we are involved. Now, these are my declarations. Now, cost of disease-modifying therapy is huge in the world. And they are even more than the cost of other drugs used in many other diseases. So we are increasing in use, more drugs. So we have to have budgets. And unfortunately, our budget is not limited. And as you can see here, most of our budget, we sent it on DMTs. About 70% we use it on DMTs. Only 30% are being used on other things. So this has led to generation of generics. And I think the world, for example, in US, there are about 250 billion generic drug industry there. So we cannot sort of hide and say we don't want generics, but at the same time, we have to rationalize to see how to evaluate these generics when it reaches to us. Is it a good generic? Is it a bad generic? Is it whatever? And plus that our other low-income countries have really no proven drug, as Maya has shared in her slides. Now, my few questions for the audience: when you have a generic, just ask yourself, what is it? Is it a simple generic? When I say a simple generic, it's basically like ibuprofen. You just make a copy of it biochemically. A complex generic is slightly different. And you have to know that the requirements for complex generics and biosimilar are different from simple. It's not only about pharmaceutical equivalence and biosimilars, but there are more stringent criteria. So complex generics are different. Interchangeable generics, they have high standards. When I say interchangeable, that means you can interchange a drug for another. There are super generics, and there is a biosimilar, and Sarah has mentioned that biosimilar is not a copy. You cannot get that synthesizing process. So you have to know something about what is required for each one, for simple, for complex, for biosimilar, for interchangeable. And the question, is this a standard copy? But are you at us are qualified to say this? We are here to know what are the processes, but are we in a place to do that? And one of the things that you can get hint is maybe get to people like FDA's or EMA or Canadian requirements. And is this listed in their confident generics? Also taking in time that, for example, Canadians sometimes approved generic drugs for outside use, and they don't allow it for internal use, although they have a good sort of regulatory agencies. And you have to know that, does your country actually have stringent generic requirement approval, or it is loose and money oriented? So that's another thing that you have to be careful with. And we physicians believe, as Maya have said, there should be not only physicians and pharmacists and pharmacoeconomists, but there should be some independent body that judges these drugs, these generics. We are for generics which have a quality. We're not here against generics. We're here to teach you how to get the best generic, the qualified generics that can use for your patients with the best evidence available there in the market. I'll give you a few examples. Now, the EMA, when it comes to complex generics, it says it's an authorization depends partly on the results of test, which is the bioequivalence, on the reference medicine, and partly on new data from clinical trials. So bioequivalence is not only is the only requirement. And that's why people, when you argue with pharmacists, they say always bioequivalence as you have listed, but it's not the only one. And you have to know this. Developing a complex generic is a challenge. An example of a complex generic is Copaxone. Copaxone is a complex generic. And you saw the trial that Maya has presented. These people, Canadian, we have one in Oman. We have a Canadian generic for Copaxone, but this company has done a trial and showed equivalence in terms not only of bio, but also on clinical equivalence. Even when your formulations that are similar in terms of psychochemical parameters, you still may have differences in kinetics. Not to say biosimilars are a completely different world. Testing general biosimilars is more complicated. They are animal-based, they are cell-based, so they cannot be copied. Minor changes in the manufacturing process can produce different post-translational medication. Therefore, at least a human study is needed. And I'm saying biosimilar. I'll show you the guidelines of the EMA, WHO, FDA biosimilars are different. So some people are more generous. So think, does your country actually have this, or they just look at bioequivalence and that's it? It's nothing. There is nothing called bioequivalence for a biosimilar drug. So you have to know that, and whoever is evaluating this from I was sitting, and this is when you make a reference drug, you do always an analytical. You just be first one or phase 0 studies, and then you do a non-clinical PK and then clinical. Whereas biosimilar development, you need the same thing because it's basically not the same drug. It's not synthesized with the same difference, and therefore you have to get clinical trials to approve it in your country, not only bioequivalence. Now, this is one of the first drug. In Oman, we have 4 drugs now. We have Copaxone, generics. We have Sinofex, and we have dimethyl fumarate, and I'll show you. So Avonex was the first one. Now, the problem with Avonex is a biosimilar. Sinofex, as Maya has said, there are trials to say that it is a substandard copy. And I was sitting with some of the pharmacists who were saying, yes, this Sinofex was approved by a committee in the Ministry of Health or whatever. And I look at and I see the studies that are not there. So I'm really suspicious about the quality of these committees and how do they evaluate these biosimilar. They're different. And they only Avonex has been in Oman for 15 years. We have 40 patients has been on the drug. 1 year introduced Sinofex, and I yet to do a study to evaluate the whole thing. But from the way that the drug was introduced, the way it was doesn't show that we have a stringent, and I suspect the whole thing. I was very pleased to hear Sarah, which is from Emirates, have a slightly different approach to this. And I congratulate her for having a different approach. And maybe this approach could be unified among the Gulf countries and maybe the MENA regions to have our own. Like, for example, the biosimilar have FDA, for example, Indonesian have their targets, their protocol for a biosimilar, how to approve biosimilar. Now, as Maya has said, this slide was presented before. The second drug, which we talk about, is dimethyl fumarate. This is the real drug or the innovator drug called Tecfidera, and this is the one that is supplied as a generic. Now, has been in Oman for more than 10 years. The real one, 29 patients on DMF, about 12 male and 17 female. Most are naive or switchers because of side effects. Generics has actually been introduced for 4 months now. And the way it was introduced is a complete switch. This one, when I looked at the orange drug for FDA or the EMA or the Canadian, I did not find this generic. And I listed, I'm here listing the generics for dimethyl fumarate, which can be been approved by the FDA, which probably will be welcoming if one of these had been, but to substitute it for a substandard copy is a problem. And when you evaluate a generic drug, you're not only looking for these side effects, because the side effects you can detect in 2-3 months, but the clinical efficacy, you cannot judge it by MRI, only from MRI. You need at least some time. You need 1 year, maybe 2 years to evaluate the MRI surrogate marker and maybe the clinical surrogate marker. This was 2 cases where I'm gonna present in about the dimethyl. Remember, we just introduced DMF about 4 months ago. This 34-year-old brain engineer, 2017, 4 weeks of left-sided numbness, MRI spine, active lesion in the spine and in the brain. Gave history 2015, right-sided numbness, which got better in 4 weeks. On examination, he had a mild left-sided weakness. Blood, including NMO, was negative, but the OCB was positive. This was his MRI, so multiple lesions. He was started on Avonex, the real Avonex. MRI brain was done in 2000, was stable, no relapse. In 2019, was admitted because he had pain and numbness and ability to control his hand and weakness. So this was a spinal relapse. So because of this relapse, his MRI was active. So we have to change his medicine. This is a suboptimal response. An indication certainly to change the medicine. So we have changed it to fingolimod. Now, the problem with this patient that we discovered that he was hepatitis B virus positive, and his ALT was 132. And we were wondering what to do with this patient because he had hepatitis B. His ALT was slightly raised. We discussed with our colleagues. So we had a problem with him. As per them, less likely to caused by fingolimod, and reassured and advised to continue. However, patients stopped taking the drug because he thought this was because of the drug. He was started on antiviral medications. We've been consulting our gastrohepatologist because of this. In 2020, he came with 1 week history of dizziness and unsteadiness, more on walking, because he stopped the drug. He was planned to start fingolimod, but the ALT was stable. MRI was no relapse. He also has a recurrent oral ulcers. And because of these oral ulcers, we stopped fingo. The ulcers really got better. And then he was challenged with fingo. He had a lot of ulcers, and therefore we had to stop it. But because there was no MRI evidence, the stable clinical, we started DMF. This was started in 2021. His ALT was stable. His MRI was stable. In March 2020, DMF was changed to generic, about early April. In May, about 2 months later, he started having a spinal relapse. He came again with a spinal relapse. And you can see the lesions in the spine and in the brain. VV was absent bilaterally. So he had a severe relapse. He had bilateral optic nerve and spinal. MRI showed a lesion in the spine. So now what we have faced is the patient only 2 months started on the generic, he was stable for almost 2 years, and he had a relapse. Now we have to switch him to natalizumab because of what happened. And we have lost this patient initially. Now, this was another case, a 29 years old. Actually, I have just seen him 2 days ago. A 29 Omani male, diagnosed MS 6 years ago. He was initially started on interferon, doing well, had radiological and clinical relapse. He was changed on fingolimod 3 years ago, doing well until 2 years, again had side effects. 2 years ago, he was started on DMF. He was stable radiologically and clinically. Now, 2 months ago, he was changed to generic medication. He saw me just actually yesterday. When I reviewed his MRI, which was done a week ago, we saw that there are more new lesions now that are coming now in his brain. So again, we have changed to this man to natalizumab. So now these are 2 patients after 29 who only 2 months after starting generic had relapsed. Is it really the generic has done it? There is substantial evidence. We cannot be sure. But yes, it looks like from substantial change in medication, they were stable for 2 years. And therefore, this was 2 patients that I presented. I haven't been able to get the 29 patients. The third drug is Gilenya and Figona. When Figona came, actually the company has presented an FDA certificate. So although when looking at the FDA site, it wasn't there. So probably it was a presentation certificate. So Figona or Gilenya, we accepted it to be started on a new medications, on a new patients, and things went on. 6 months ago or 9 months ago, I did a study to compare the 2 generics, and you can see the Gilenya and the Figona. There are about 26 patients now, maybe more. Female 52, male 48. The age about 36. The age range up to 67. The mean duration of treatment is about 16 months. The EDSS score is about 1.9 months. Now, let's look at the results of the Figona now. There was 80% same, no change. There was 20% increase in the T2 lesion in the MRI. GAD, there was 10% show enhancement. 20% had relapse. 90% in EDSS had no change, but 10% had an increase. Now, when we compared it to actually the generic, the original drug, the Gilenya, we found a similar annualized relapse rate. So annualized relapse rate was 0.16. But the EDSS, 19% did not progress on fingolimod, or 10% did not progress on fingolimod, 19% progressed on the Figona. So there is a difference in the EDSS progression between the generic and the brand. Side effects, away in comparison, we have seen some more elevation in liver enzymes, more thyroid issues with the generic as compared to the Gilenya. So this was my experience with at least 3 drugs that that have been introduced in Oman, and Copaxone is the one which is introduced. But Copaxone maybe have a different story, but I think it got a Canadian approval, and it was a trial comparing Copaxone with the original drug, and that has shown similarities. I'm happy to take on, for example, Copaxone, but I'm questionable about the other 3s. And that's my last questions, my last presentation, and I'm open for any questions that anybody has. Thank you.
Thank you so much, Dr. Jaber. It was really important to end with the real-world experience, and you really offered us very practical tips from your own personal experience at your center in Oman. So we would really like to thank you for that. We would also want to remind our attendees to fill the evaluation form, which is shared right now in the chat box. Now let's take some questions. One of our attendees is asking, why would we use generics if they are not more cost effective than innovators?
Now that's a big topic. And yes, some of them are not cost effective and we should not be using, but some of them are. So, and the branded one, the one that we trust, some of them have cost effectiveness. So we cannot just blindly say they are not cost effective. And at least some are cost effective, and we should be welcoming, provided that they have a good generic, not a substandard generic, and they are approved in an appropriate way.
Exactly. We have another question. It's in respect to one of the cases. I believe the second case, but maybe we can get clarification from the person asking the question. They're asking that the patient had poor prognostic factors and whether they should be on high efficacy DMT. I think this was the second case, but I'm not 100% sure.
The spine lesion may have been a poor prognostic factors. But you know, people, sorry, neurologists have different opinion, and some of them would not. So he was under another consultant who was started him on Avonex first, and then came on DMF. But as we all studies have shown, that the earliest you start your patient, less than 1 year, they have the best effect. Now, even in England, there's a study to say that we should be starting them within 3 months. There is ongoing study now to see. So there are now world evidence-based that you should starting your patient, whatever. And now there are much more data saying that the high efficacy drugs are doing better than low efficacy drugs. So in answering, yes, I would agree that if any patient with MS who has poor prognostic signs, we should probably more. Not only that, but there is a trend now among neurologists and MS physicians is that when we had low efficacy drugs, we were worrying about safety, cost, but over the years, now these things it's not standing, and we now have drugs which have come in the market, which are more effective with less side effects. So we now are really moving, for example, to maybe thinking about maybe we should be starting more patients, even those with not many poor or hardly poor prognostic factors on high efficacy drugs, because they are safe and they are now less costly than we thought before. Exactly.
Because we have to look at the big picture of the cost, as you said, and the cost over the lifespan of the patient, which is probably changes our treatment methods and towards starting more high efficacy drugs, as you said from the beginning. So another question is, what if an MS patient is doing well on generic DMF? Should we continue?
Well, as I said, the first thing is to make sure this generic is real generic, not a substandard copy. This first thing. Yes, if the patient is doing on a good generic, doing well, and he is doing so, we should not be taking off, and we should continue. But taking into your mind that in time will only tell. So you have to separate these patients, see what happens in 1 or 2 years. Look at their MRI, look at the clinical data, and compare them with the group that you have on. If they are doing well, then well and good, and you should continue their patients. And only time will tell. But initially, you need an approved good generic as opposed to substandard generic to make this move and compare the two drugs.
Yes, to be confident in your move as well. Another question is, can we consider using generics from companies that have US FDA approval?
I think if the generic, because we trust FDA, when we submit a new drug to even our regulatory authorities in the Gulf, what do they require? They require an FDA approval. They require an EMA approval. Sometimes some of them require NICE approval. So they trust the new drug on these 3 criteria, and cost effective, obviously. But if a drug has an FDA EMA approval, then I think we should trust the drug. But yeah, there's one catch only, and I alluded to in the Canadian part, because some of these approved a generic, and this generic is not used in their countries. So what you have to be a little bit be careful about this kind of behavior.
Yes, so like look at the data appropriately and look at the usage of these generics, where they are used, in which countries, as you said, and whether the countries have approved them or not. So I think these are all the questions. I would like wrap up with a patient-centered maybe question and ask you if your patient has a fear from using generics, do you think their fear is substantiated? How would you address this concern in a very patient-centered approach?
This was a good question. As there was I was sitting yesterday in the MS clinic, and this young patient came, and he was probably anxious and said to me, look, they gave me a different generic, the DMF, the new generic, and I'm afraid I will get a relapse, and they will not change the issue. Then I explained for him. I have to explain for him in all what is this generic, what I know about this generic, whether I as a physician trust these generics or I don't trust these generics. And we have to be frank with our patients of what we know. We're not here to advocate companies or ministries or anything. We are here to tell an independent physician opinion to our patients. This is really what we call amana in our, and we physicians are the corner of our patients. They trust. They don't look at the pharmacist. They will not blame the pharmacist. They will blame us physicians. And we have to be confident with them and tell them. Obviously, if I trust that generic, then I will tell him, look, this generic is good. This generic has been approved in the US or in Canada, or the patient might get convinced that this generic is a well-trusted generic, and therefore he might take it. But if I'm not myself convinced with this generic, I can't convince my patients with such a generic.
Exactly. So we should let the data really drive us to be able to give our patients information so that they are able to make themselves informed decisions as well.
Yes, yes. And in fact, he told me, I don't want this. I changed my drug. Yeah.
And here the patient autonomy really plays maybe a role or drives us in making these decisions as well. And all of us as healthcare professionals to be able to give the patient the best advice possible, as you said, based on the evidence. So I think we have one more question. We'll take that as well to end. How is the average compliance with innovative DMTs? And in your practice, how many years patients are taking or maybe staying on the drug, I believe, complying?
So as Maya have said, there are 19 drugs. So the average compliance with the drugs depends on its infusion. Infusion has a better because you give it monthly and it can reach up to 90%. The orals, the drugs, for example, the interference with the injections, the compliance is really low. So you can get up to 40% or even less. For all drugs, maybe in between. So you get a difference between different formulations, different drugs. And so you can see between 40 and 90%, depends on the route of administration, depends on the drugs. So it's like depends on the number of drugs, number of patients, a number of how many orals in a day, as opposed to PID, TID, or OD.
So the range is a lot. It depends, as you said, between all the DMTs, and we have a huge landscape of DMTs to look at.
Yes.
So I believe with this, we will end your talk. Thank you so much, Dr. Jaber. It was a really great way, as I said, to wrap up this interesting webinar today. Thank you.
Thank you. Thank you all.
Okay, so if we don't have any more questions, I will thank everyone for attending today. First, I'd like to thank our faculty, Dr. Sara Dallal, Dr. Maya Zainuddin, and Dr. Jaber Al-Khabouri. Thank you to all our attendees for sticking with us till this time. We finished perfectly on time. So we reminded you to fill the survey, and we'd also like to inform you that the certificate of attendance will be sent by email within around 1 week from today. So you will receive your certificate of attendance. We would like to reassure you as well that if you missed any part of this webinar or you are interested to revisit the lectures, you can check our IVPN Neuropsychiatry library, where we always post all the recordings. We would also want to remind you to join our IVPN Neuropsychiatry network and follow us on all our social media platforms to learn about our upcoming activities. I'd like to thank Dr. Maya for her leadership on the IVPN Neuropsychiatry listserv, and we want to remind you that we will have an upcoming activity on July 5, centered around the management of MS. Please join us and stay tuned for the announcements. It will be a very interesting talk as well. In the end, I'd like to thank everyone for attending, and definitely I would like to thank our sponsor, Biogen, for supporting this educational activity and really helping us give our patients more quality care based on all the evidence provided by our faculty. Thank you, everyone, and see you on July 5.